Hello. For this note, we are only going to do part of the cell cycle notes at first, and we'll do the rest in a separate video. So if you're taking notes along with this video, you should only be completing part of that note sheet. So let's start with, since we are talking about the cell cycle, we need to talk about DNA because DNA is very, very, very important for what happens in a cell's lifetime. So to start out, what is a chromosome? What even is that? You may have heard that word before, or you may not have. But if you haven't ever heard the word chromosome before, what it is a single DNA molecule. So DNA, deoxyribonucleic acid, that is a really, really, really long organic molecule, as we just learned. And if you take that DNA molecule that's really, really, really super, super, super long, and you wind it up, like you twist it up and twist it up and twist it up. If you've ever twisted a rubber band up until it starts knotting in on itself, that's what I'm talking about. So you take that and you twist it and you twist it, and you twist it around these little yellow balls over here, and they're called histones. So like this little yellow thing here, here, and here, those are histones. You twist it around that and you put it all together and twist it up and twist it up and twist it up more. And eventually around the end, you get these things called chromosomes, which are super, super twisted DNA clumps. So chromosomes are made of DNA, as I already stated, but also proteins. That's what those histones are. They are protein. And all of that together is found in eukaryotic cells. Now, if you've never heard the term eukaryotic before, we are made of eukaryotic cells. Eukaryotic cells are cells that are found in really complex organisms. So any plants are made of eukaryotic cells. Any animals are made of eukaryotic cells. So chromosomes, those big clumps of DNA and proteins, are found in eukaryotic cells, and they only are found during cell division, when a cell divides. Okay, so what does a chromosome look like during cell reproduction? Well, there are going to be two halves to chromosomes, right? So if we look at this picture down here, we see one half right here that I'm outlining in red, and the other half that I'm going to go ahead and outline in blue. Okay? And then right in the middle, right here-ish, we have this structure that links those two halves together. You can kind of see it a little bit better over here. And that is going to be called a centromere. They are going to form when DNA makes a copy of itself before cell division. So let's pretend for a second that you just have this big blob of kind of unwound DNA hanging out in the nucleus of a cell. That's what DNA is going to be like most of the time. But if a cell is going to divide into two, what needs to happen is all of that DNA needs to make a copy of itself. And a big mess of unwound DNA is not very easy to copy. So the cell is going to wind it up and wind it up and wind it up into a chromosome. And as it's copying it, the other half is going to form on the other side. And then those two things are connected with that centromere. So you see that starting to look like this situation over here. So that centromere really conjoins the original DNA segment with the copy DNA segment, is what I'm saying to you. Okay? So there's a couple pictures that I would like you to look at. Some of them are diagrams, some of them are actual photos using electron microscopy. So that's what these are up here. This middle photo here is chromosomes under a scanning electron microscope. This one over here is through a light microscope. Light microscopes, by the way, are the same type of microscopes that we would use in a normal science classroom. You've used a light microscope before. The only difference between the ones you use and the ones that are used to picture these chromosomes is they can zoom in a little farther. They're a little bit nicer, but on the whole, same. This is a diagram of a singular chromosome down here. So before all of that DNA makes a copy of itself, after the DNA makes a copy of itself, and then after those cells divide, you'll end up with a singular copy of a chromosome around the end, okay? So how many chromosomes do we have in us? Because we don't have just one. Each species of animal has a unique number of chromosomes in each of their cells. So for humans, we have 23 pairs of chromosomes in our body, 46 total. So you notice down here, we have 46 total chromosomes. So for chromosome 1, we would have one copy of chromosome 1 that comes from mom, and we would have one copy of chromosome 1 that comes from dad. And then for chromosome 2, once again, we would have one copy from mom, and we would have one copy from dad, and so on and so forth. And so you can see how if we have 46 total chromosomes, because this is one chromosome, that's one chromosome, but they do come in pairs. So chromosome 1 comes in a pair of 2, chromosome 2 comes in a pair of 2, so on and so forth. Pea plants are going to have 14 chromosomes, or 7 pairs. Sunflowers are going to have 34 chromosomes, or 17 pairs. Cats are going to have 38 chromosomes. Pufferfish, 42. Humans, we have 46, or 23 pairs. Dogs actually have 78 chromosomes. So what becomes pretty imminently obvious is that more chromosomes does not mean higher level organism. Dogs have more chromosomes than we do, but obviously dogs are not capable of higher level calculus or any of the things that humans are capable of cognitively. So more chromosomes doesn't mean a better organism, like a smarter organism. It just is. Some relatively simple organisms have hundreds of chromosomes. It just kind of depends on your species. It doesn't mean anything necessarily as to how smart that organism is or how strong. How are human and animal chromosomes categorized? So we're going to categorize them in a few different ways. So if this is one chromosome, we are going to categorize those chromosomes by how big they are. There are going to be some banding patterns, which I can show you at some point, but basically chromosomes have little bands on them, and you can see them if you're looking closely enough. And each banding pattern is going to be unique to which chromosome we're talking about. And then the third way that we categorize them is the position of the centromere. So again, the centromere is here and here in this little diagram. Some chromosomes look very, very similar, but their centromeres are in a different position. So that's one way we can tell the difference. This over here, this picture right here, is called a karyotype. It's not important that you know what it's called right now, but what a karyotype is all of the chromosomes laid out and organized by pairs. So you can see chromosome 1, 2, 3, 4, 5, 6, 7, blah, 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 all the way down to 22. And our 23rd pair are our sex chromosomes. So if you've ever heard that girls are XX and boys are XY, that is actually referring to the last pair of chromosomes that we have. If you have 2 X chromosomes, you are female genetically, and if you have one of each, like this particular karyotype has 1 X and 1 Y, then you are male genetically. So what's important about those sex chromosomes is that they determine your biological sex, which determines traits that you will have when you're born and traits that you will have as you grow. It's not always traits directly related to reproduction, though. So a lot of people assume that because they're the sex chromosomes, they only carry genes for your sex organs or whatever, but that's not true. That's not true at all. Color blindness is something that is a sex-linked trait, and by that, what I mean by that is that when you are colorblind, that means that there is a gene on your X chromosome that is deficient in some way. So boys actually are far more likely to be colorblind because they only have one version of their X chromosome, whereas girls have two versions of their X chromosome. So essentially, girls have another chance to have a good workable copy of that gene, whereas boys only have one shot. And if their copy that they get is defective, then they are colorblind. Autosomes are effectively everything else other than your sex chromosomes. So chromosomes 1 through 22 are going to be your autosomes. So that's all of these that are in the purple section up here. Those autosomes are going to code for every single other trait that you have besides the ones that are coded for by your sex chromosomes. And it's a ton of stuff, you guys. It's a ridiculous amount of information is encoded for on these chromosomes. What are homologous chromosomes? So if we break apart that word like I so love to do, the prefix homo means same. So the word homologous refers to the same chromosome. So every organism that is produced by sexual reproduction, so that's humans and animals, and also plants, some plants, not all, but some. Every single one of those organisms are going to have two copies of each of your autosomes, okay? So remember how I was saying you have one chromosome, one from mom, and one chromosome, one from dad, and one chromosome, two from mom, and one chromosome, two from dad. These are homologous. They are the same chromosome. It's just that one you got from one parent, whereas the other you got from the other parent. So if I say the word homologous chromosomes, you should think of one of each from each parent. One chromosome, one from mom, one chromosome, one from dad. That's what you should think of. So you can say that a couple of ways. You can say that the two chromosome ones are homologous to one another, or you can call them homologs. It doesn't really matter which one you choose. They both mean the same thing. But the biggest thing to know is that each homologous chromosome is going to be the same size, the same shape, and carry genes for the same trait. So once again, referring back to chromosome 1, let's pretend that these are both the same size because I am terrible at drawing. The copy of chromosome 1 you got from mom maybe has a gene for eye color, and the chromosome 1 that you got from dad has that same gene for eye color in the same place. They may not say the same thing, like maybe this one says you're going to have brown eyes, and that one says you should have blue eyes, in which case, due to information that I will teach you later, you might have something else, like you have a bunch of genes that control the same thing, but regardless, these are the same gene for the same trait. They don't necessarily say the same thing, but they do exist and they are homologous to one another. The next thing we have to talk about is diploid versus haploid. So diploid cells are cells that have both members of each homologous pair. So in one cell, like let's pretend that this is a cell, in the nucleus of that cell, you're going to have your chromosome 1s, your chromosome 2s, your chromosome 3s, and your chromosome 4s and 5s and 6 and so on and so forth. In your diploid cells, these cells have both members of each chromosome, okay? Both members of each homologous pair. And so they are diploid because they have both. If we break down that word, di means two. So they have two of each pair. All of your body cells are going to be diploid. The cells in your arm, the cells on your face, the cells in your elbow, wherever, all of your main body cells are going to have diploid numbers of chromosomes. They're going to have both homologous pairs. Another way to refer to diploid is saying that those cells are 2N. What N means is how many chromosomes, how many unique chromosomes you have. So if we have 23 pairs, N equals 23, but our diploid cells are going to have 46 chromosomes because they have 2 copies of each one, okay? But haploid cells, on the other hand, are cells that only contain 1 of each pair of chromosomes. So whereas a diploid cell over here had 2 of chromosome 1, 2 of chromosome 2, 2 of chromosome 3, so on and so forth, your haploid cells are only going to have 1 of chromosome 1, 1 of chromosome 2, 1 of chromosome 3, and so on and so forth. Those are your sex cells. Those are your sperm cells and your egg cells. And the reason that they exist in that manner is because they have to. Because imagine this is a little sperm cell. It's going to go meet up with an egg cell that has 1 of each as well. If both the sperm and the egg cell had a full 46 chromosomes, the child resulting would have 92 chromosomes. And that's not good. More chromosomes is not necessarily better. If you have too many copies of chromosomes, you might end up with a lot of problems, a lot of disorders, and sometimes if you have too many copies of the wrong one, it's actually fatal before you're born. So that can cause miscarriages and a whole lot of other things. So to combat this, each sperm and egg cell have half the number of chromosomes, because if you add 23 plus 23, you get 46, which is a normal human cell. And so we say that haploid cells are 1N because N equals 23, because these are only going to have 23 total chromosomes, whereas a diploid cell over here would have 46. What is the cell cycle? So here I've explained chromosomes and I've explained a little bit about them. So now we need to talk about what the actual cell cycle is, meaning the set of steps that makes up the life of a cell. Because just like you are alive, your cells are alive and part of you. So we use this diagram, this big circle diagram, to kind of explain the life of a cell. So way over here, this is kind of like the start line. A new cell is born, effectively. And then it's going to go in this direction, okay? Through the G1 phase, the cell is just growing. It's growing, it's doing its thing. And then once it hits the S phase over here, it's almost going to be time for the cell to divide again. So the cell is going to replicate all of its DNA that's just hanging out in the middle. Then after it finishes replicating the DNA, that cell is going to prepare to divide again. And then in this little pie chart section is where we have mitosis. And mitosis is the division of the nucleus. So basically, our cell is going to prepare its DNA and divide its DNA, and then after all the DNA is divided up, then in this tiny little section right here, the rest of the cell divides. Your cytoplasm, the rest of the cell, effectively. We also call this cytokinesis. So if you ever hear the term cytokinesis, it means that the cell itself is dividing there. Mitosis is the nucleus dividing. Cytokinesis is the rest of the cell dividing. Okay, so we can break down mitosis into a few different things. But on the whole, mitosis, again, the division of the nucleus, it's going to result in 2 new cells. the genetic material of those two new cells is going to be identical to the original parent cell. And that is important because let's say you cut open your arm on accident, your body has to repair that damage. And it does this by making new cells. So if those cells were different to your original cells, that would be a problem, because then your body's going to be like, oh my God, foreign cells, and then mark them for destruction, and you would reject the new scar tissue, and it would be a whole mess. So most important thing is that mitosis results in genetically identical cells to the parent cell. Okay? This here, let's see if I can click on it and play it for you guys in video. Whoop, it did the thing. Oh no, the video is unavailable. That's unfortunate. Well, that's okay. We will watch this in class together then. So we'll have to watch that later. This is a pretty cool animation of cell division. You can watch it happen, but again, we'll watch that in class together. But like I said before, we can break apart cell division into different phases. The first phase of cell division is called prophase. And a few things happen in that first phase. Chromatin is another word for DNA. So what's happening is your DNA is thickening and condensing into chromosomes in this phase. Your nucleus is going to disappear in this phase. Something called a centrosome, which is a part of a cell, is going to move to the opposite poles of the cell. And then spindle fibers are going to form, and they are going to want to eventually attach themselves to the chromosomes to kind of pull the extra chromosomes apart. So right now, if I were to just draw a little doodle of what's happening, here's your nucleus that's kind of breaking down a little bit. Here's your chromosomes that are forming in the middle. Here's your little centrosomes that are moving to opposite ends of the cell. And here's your little spindle fibers that are, there's the bell, starting to form at that end. Okay, so this would be a good doodle of what's going on right there. The next phase of mitosis is metaphase, and I have middle in parentheses because all those chromosomes are going to go ahead and line up in the middle of the cell for metaphase. Okay, so once again, if I wanted to draw a doodle of what's going on, all those chromosomes are going to go ahead and line up in the center of the cell, and all of those spindle fibers are kind of attaching to those chromosomes. Okay, we have the little doohickeys over there. So this would be a good representation of what a cell looks like if I were to doodle it out. Again, you can see that in an actual cell over here. You can see all those chromosomes lining up in the middle. In anaphase, which is the very next phase, chromatids are going to separate. Sister chromatids separate. So meaning those two copies of each chromosome are going to be pulled apart by those spindle fibers, okay? So once again, I'm going to doodle it out. We have these little guys with the spindle fibers over here. And they are pulling those chromosomes apart. Okay, so we see those chromosomes moving towards either end of that cell. The last phase of mitosis, known as telophase, is kind of the last part of mitosis. So what you see happening is the cell is kind of starting to look like two cells now that are just barely connected in the middle. You see the chromosomes still kind of being pulled outward a little bit, but you see this nuclear and cell membrane furrow starting to form here and here, which eventually is going to pinch off the cells into two. Okay? Another thing that you'll notice happening is that the spindle fibers are going away, and also these chromosomes that are still being pulled in either direction, as soon as that nuclear membrane starts to reform around them, they're going to start to uncondense themselves back into chromosomes. Or back out of chromosomes, excuse me. After that happens, we have cytokinesis, which is the final division of the rest of the cell. Here is a very nice doodle for you to kind of show you the phases. Whoop. Right here. This here is actually interphase. This is the normal cell phase. You see that the DNA is all uncondensed in the middle here. This is going to be prophase, where you see the nuclear membrane start to disappear and the condensation of your chromosomes happening. This is going to be metaphase, where you see those chromosomes lining up in the middle. This is anaphase, where you see them being pulled to either end. And this is telophase, where you see those nuclear membranes start to reform and the DNA start to uncondense. So to talk about cytokinesis a little bit more, as I already stated, it is the division of the cytoplasm, or the rest of the cell. Cytokinesis is going to begin during telophase. So as you can see down here in this picture, that little cleavage furrow, or that little pinching off that you see happening here and here, that is the start of cytokinesis, okay? In plant cells, you don't really see the same thing because plant cells have a cell wall around them. So instead, you're going to see the formation of a cell plate in the middle that's going to separate those two cells. But either way, you're going to get identical daughter cells out of it, either animal or plant daughter cells, but they will be separate cells. Each of these cells have about half of the amount of cytoplasm of the first original cell and about half of the organelles, because those organelles are going to divide pretty evenly between the two. So if we were to go ahead and label one of these diagrams, like I would absolutely ask you to do, we have these centrosomes that are going to be right here. Then next we have the chromosomes that are going to start to condense there, your DNA. After that, we're going to notice our spindle fibers forming there. Then we were going to label the phases. So this phase with the uncondensed DNA, the uncondensed chromosomes, that's going to be interphase. Where you start seeing that condensation is going to be prophase. Metaphase is going to be when those chromosomes line up in the middle. Anaphase is when they start to pull apart. Telophase is when that furrow starts to form. And finally, cytokinesis is the final division. This would be something that I would star or draw or box or know for myself, because this is the kind of diagram I might ask you about on a test. And this is the point that we are going to go ahead and stop for now. Hopefully you got these notes with me in class, but if you were absent, this would be a very good video to watch.